nuclear division involved in this? And what is it? What kind of nuclear division are we talking about? Formation of gametes. Mitosis or meiosis? Meiosis, okay? And the key feature of meiosis is what? Reducing the genetic material by 1/2, reducing the chromosome number by 1/2. That's why it's sometimes called reduction division. The point is you want to form cells, gametes, that are haploid. What does haploid mean? They have single set of chromosomes, okay? They're not paired, the chromosomes are single. Our normal body cells are diploid. What does that mean? Each chromosomes are in pairs, okay? And diploid is usually referred to as 2n, usually lowercase n, with n being the number of chromosomes. What's a 2n number for humans? In other words, how many chromosomes do our normal body cells have? 46, in 23 pairs. The gametes then, being haploid, only have 23 single chromosomes, not paired. When the two gametes come together in fertilization, you restore the diploid cell. That is the fertilized egg or zygote. That's the whole point of this. Reduce the chromosome number by 1/2, and the two cells with half of the chromosomes come together, sperm and egg, they form the fertilized egg or zygote. That is diploid, and all the division from then on is going to be by mitosis. Because mitosis maintains the chromosome number. The daughter cell nuclei have the same number and kind of chromosomes as the driving cell. So most of the division we see in the cells in our body is involved mitosis. From the embryo on, that fertilized egg, right up through our own bodies now, and the tissue repair that goes on throughout life also involves mitosis. You want cells just like the cell that's being replaced. Okay? You got to have a full chromosome complement. It's only when you're forming the gametes, it's from an egg, that we resort to meiosis, because those haploid cells, the gametes. Okay, so the process of spermatogenesis is the formation of these gametes. In males, it's called spermatogenesis, because you're forming sperm cells. In females, it's called oogenesis, because egg cells are being formed. The process is very similar. Okay, it's illustrated here, what I call these bubble charts. As I said, we were going to slides last time. You do not need to identify the individual spermatogenic cells. Those are cells directly involved in spermatogenesis on the slides. Okay? But you do on this. Okay? And it shows on one side spermatogenesis, on the other is oogenesis, and the stage is in between the formation, the final product of the gametes. Okay? But I want you to know three things. I want you to know the cell, the particular cell here, by name. I want you to know if it's haploid or diploid. And if we're a human cell, which these are not, how many chromosomes do you expect to see in that cell? Okay? So let's go over this. Again, you're responsible for the names of these cells, whether they're haploid or diploid, and how many chromosomes, were they human? Okay? Okay, ignore this. All right, this is declaration. Where does this occur, spermatogenesis, in the male, specifically? Where? No. Testes, okay, but be more specific. Where are the testes? The wall of the seminiferous tubules. This process going on in the wall of the seminiferous tubules. That's where the sperm is formed. What occurs in the epididymis? Which picks up the sperm from the testes. It's stored there temporarily, and what else? It matures there. Okay? So epididymis, temporary sperm storage, and site of sperm cell maturation. The actual sperm cell formation is in the wall of the seminiferous tubules. That's where this process occurs. Okay? All right, we start. This is all labeled, okay? But obviously, you will see this again, not the labels. You'll apply the labels. All right. We're going to start here with spermatogenesis. That's the process of sperm cell formation. These represent the sperm cells at the end of the pathway. As I say, ignore these. You do not have egg cells in the testes. All right. We start with this cell. This cell is diploid. It's the precursor cell. It's called a spermatogonium. It's indicated by 2N, which means diploid. Okay? This is a precursor cell. It's a body cell, like a typical body cell. It's got pairs of chromosomes, 2M. Again, this is all in the wall center posterior. This cell undergoes mitosis, not meiosis. Mitosis to produce daughter cells, okay? One will be a replacement for this. The other is going to be what's called a primary spermatocyte. It too is diploid because mitosis, remember, maintains a chromosome number. You start with a diploid cell, your daughter cells are going to be diploid. Okay? Like I say, this reproduces 2 daughter cells. One will be another one of these. Okay, so you keep this process going. The other one's going to be a primary spermatocyte. All diploid because the division here is by mitosis. You maintain the kind of number of chromosomes. It's this cell, the primary spermatocyte, that undergoes meiosis 1, the first meiotic division. It produces 2 daughter cells. Okay, this is the intermittent stage here showing anaphase. Here are the 2 daughter cells. One's already going through meiosis 2, and these are called secondary spermatocytes. And they are haploid. So at the end of meiosis 1, you've got haploid daughter cells. In this case, they're called secondary spermatocytes. And by the way, the number with the degree symbol, number 1, the degree symbol means primary. Number 2, the degree symbol means secondary. Okay? Now what's happening in meiosis 1? The homologous chromosome pairs, which are lined up side by side, in metaphase simply separate one from the other and go to opposite poles. So you've got these chromosomes, okay, but not only half what you started with. What do we start with up here? Something chromosomes in this cell. It's a diploid cell. Everybody see it? Purple things are chromosomes? Count them. What do you got? How many do you see there? 1, 2, 3, 4, 5, 6. So 2n equals 6 here. All right? This for a human cell, diploid, how many chromosomes? 2n would equal 46. Okay? That's what I said. Right? All right, we got 6 chromosomes here, and they're paired. Down here, how many chromosomes do we have? How many chromosomes is that cell? Count them. 3. We started with 6, so we've reduced the chromosome number by 1.5. These are diploid cells. Now, don't confuse double-stranded chromosomes and single-stranded chromosomes with diploid and haploid. You may have diploid cells with single-stranded chromosomes. Or you may have haploid cells with double-stranded chromosomes, like you see here. It's whether or not the chromosomes are in pairs. If they're in pairs, double or single-stranded, diploid. If they're not, double or single-stranded, haploid. That's what's going to happen here. Where the cell divides, it's going to replicate its DNA when we become double-stranded chromosomes. That's the key here. Okay? All right. These cells, secondary spermatocytes, undergo meiosis 2. As I pointed out in lecture, meiosis 2 really involves just simply splitting double-stranded chromosomes into single-stranded chromosomes. You don't change the chromosome number. So the daughter cells from this division, meiosis 2, are called spermatids. And they got the same number of chromosomes as the secondary spermatocytes. The only difference is they're single-stranded now instead of double-stranded. So these two are all haploid cells. Okay? These cells then produce the sperm cells. What kind of division is involved here? Any guesses? Mitosis or meiosis? I would call that a trick question. Look, for each spermatid, you get one sperm cell. No division. It's simply a maturation process. It's called spermatogenesis. Okay? We haven't covered yet lecture one today. Okay? This last step here, going from spermatid, a haploid cell, to a sperm cell, also haploid. It's 1 for 1. No division. It's maturation. And that involves 3 things. The obvious one is development of the flagellum. So the sperm can be motor. That's one thing that's involved. A second thing, and it doesn't really show well here, it's loss of most of the cytoplasm. You want the sperm cell to be compact. It's easier to move a small cell than a big one. The egg can be big. You want to keep all the cytoplasm, not the sperm cell. It's simply a vehicle to get the male chromosomes into the egg. It's a lot easier to move this small, compact sperm cell, which is not to scale, by the way, if it's tight. And that's why most of the sperm cell okay, has very little cytoplasm. So that's the second thing, shedding excess cytoplasm. Form a flagella, shed excess cytoplasm, and then, and of course it goes during the model, development of what's called an acrosome. I'll talk about that lecture today. Okay? Acrosome is at the head of the sperm here. It's a little bag of digestive enzymes, much like a lysosome. Why? Because the sperm literally has to digest its way through the barriers from the egg cell to get the egg cell cytoplasm to accomplish fertilization. That's why sometimes it needs help with its buddies, more enzymes. Okay? That's all a maturation process. Development of the flagella, shedding excess cytoplasm, make the sperm cell very compact. Again, this is not to scale, unfortunately. And then, from that acrosome, a little bag of digestive enzymes. Okay? We'll talk about this in lecture also. And notice it's one for one. And notice, we started with a precursor cell, diploid, mitogonum, and how many gametes do we get? 4. It's an ongoing process. That's why the sperm cells are produced by the millions. A typical ejaculated human male, healthy, something over maybe 20 million sperm cells. It only takes one for fertilization. That's because most of them aren't going to make it. Okay? It's a long swim. Only the best survive. Like I say, sperm is deposited where in fertilization or intercourse? The vagina. Okay? You don't tell me anything new. Right? Now where does sperm have to go? Got to swim up the vagina, the uterine cavity, and then one up the fallopian tube. Which one? There are no road signs. Like I said, males don't ask directions, especially females. Right? We're going to choose one. Right? You choose the wrong one, it's a dead end. No egg. Okay? And then you've still got a long way to go to get up that fallopian tube to greet the egg. Because usually fertilization, the upper third or so of the fallopian tube. That sperm really has a long distance to go. Got to be taken swimming, okay, and got to get up there intact. Yes?
Does not the egg release some kind of chemical scent that the sperm would be attracted to?
Is there a chemical scent?
Well, yeah, I mean, I thought I had heard that maybe.
Well, it's not really a scent.